Today's topic is formed elements. Formed elements in blood. Where it is, these elements are presented in the blood. We know that the blood is composed by the aqueous portion, elemental portion here. The aqueous portion is nothing but as a plasma and their composition. That means the number of minerals, vitamins, proteins, water, so pigments, etc. They comes under the plasma. And the elements. Elements means these are nothing but as a cells here. Different types of the cells are presented. Different types of elements are presented in the blood we are taking. Generally, the elements are called as a corpuscles. These are called as a corpuscles or blood corpuscles here. Commonly called as a blood corpuscle. These formed elements, how many types of the formed elements are, how many types of the corpuscles are presented means majorly we are differentiate the three types of the elements are presented. So what are these three types of the elements in the blood means one is erythrocytes here, second one is leukocytes, third one is platelets. These are the three types of the elements. Three types of the cells are presented in the blood we are taking. Erythrocytes, commonly called as a RBC. Leukocytes, commonly called as a WBC. And platelets are thrombocytes here. Okay, platelets are the thrombocytes. So these are the three types of the elements are presented in the blood along with the plasma we are taking. And their important, their shape and their structure, their function one by one we can be going to discuss. First one is the RBC. RBC. Red blood cells here. Red. So the name itself is explained erythro. Erythros means red in the color. These are the erythro. So means red color. So these cells are red in the color. So that only name given the erythrocytes here. So when we are taking into the erythrocytes, RBC, the total number of the blood corpuscles, these are into the major content they are presented into the blood we are taking means. So how many number of RBC cells are presented in a blood, human blood we are taking means 5.5 to 5 millions in case of the male per 1 cubic meter of the blood sample we are taking. In case of the female, 4.5 to 5 millions female, so per 1 cubic meter of the blood sample we are taking. So these many number of erythrocytes are presented in the 1 cubic millimeter of the blood sample we are taking. So that only these are the abundant. Then we are taking into the structure of the RBC. Here the erythrocytes are surrounded biconcave. These are the biconcave. Circular, round, surroundly are the circular, biconcave. Biconcave means their margins are becomes into the elevated, the center becomes into the concave structure. So biconcave cells, these are circular cells here. These are generally having microns here. The diameter of the RBC we are taking is 7 microns. Okay, so 7 microns, millimicrons diameter they are having. So why they becomes into the biconcave structure means to increasing into the surface area. The surface area of the RBC, to increase the surface area of the RBC, these are forming in the biconcave structure here. So biconcave structure. So whether you are taking the cell structure of the RBC, here the plasma or the cytoplasm of the RBC, it is dissolved, a pigment is called as a hemoglobin. The hemoglobin is dissolved form in the cytoplasm of the RBC, so that only the RBC cells, they becomes into the red in the color. Hemoglobin is nothing but as it is a complexed molecule forming by the heme and globin here. Heme is nothing but as a iron molecules. Globin is nothing but as a protein molecules. Okay, with the conjugate protein. This conjugate protein in the dissolved state, the RBC cytoplasm is having the cytoplasm is dissolved into the hemoglobin. So that only RBC we are taking, so it is a red in the color we are taking. And why it is forming the biconcave structure means to increase the surface area. So here to increase the surface area. So how it is increased the surface area means early in the developmental stages, the nucleus, mitochondria, Golgi complexes, other cell or endoplasmic reticulum, these are presented. Later on we are taking into the RBC, these cell organelles are absent. Why they becomes absent means more amount of hemoglobin when it is presented, the hemoglobin more amount is presented, the cells are carrying the more amount of the oxygen, because the hemoglobin is binds with the oxygen and carbon dioxide here. That means exchanging of the gases, transportation of the gases, exchanging and the transportation of the gases is conducting by the hemoglobin. Actually, the hemoglobin is diffusing intracellular fluids, inter and the intracellular. So between into the inter and the intracellular, that means outside the fluids, intercellular fluids, inside the intracellular. Between into these two fluids, gases are exchanges, or the materials are going to diffuses with the help of the, so in this hemoglobin, gases are going to exchanging here. So that is the major function of the hemoglobin. This hemoglobin is presented in the dissolved state of the cytoplasm of the RBC. So like this, whether we are taking into the structure, like this structure is presented here. Then RBC major function we are taking means, so this is the structure here. The major function of the RBC is exchanging of the gases. Exchange of gases we are taking. So where these are going to exchanging means the oxygen and the carbon dioxide, they are supplying the, so from the different parts of the body, from the different parts of the body to the lungs to the different parts of the body, oxygen and the carbon dioxide is going to exchanging, transporting and exchanging into the different cellular surfaces with the help of the RBC only. So whether we are taking into the RBC is having into the 120 days of lifespan here. That means they are conducting this transportation and exchanging of the gases for the 120 days. 120 days of the lifespan they are having. So after these 120 days, they are going to destroyed. So that means the RBC lifespan, after completion of the lifespan of the 120 days, they are going to destroyed into the spleen. So they are going to destroyed into the spleen. So the spleen is act as a graveyard here. So all these cellular components are going to destroy. The remaining iron material, already we discussed the hemoglobin, within the hemoglobin, this So in this molecule, heme is presented and the globin is presented. Globin is in the protein molecule, heme is into the iron component here. So this heme or iron component, iron material, iron molecules are again they are going to bone marrow here. Bone marrow. To reuse. Again to producing into the hemoglobin. So like this, after the completion of their lifespan, these are going to destroyed into the spleen. The spleen is called as a graveyard here. So this is recording to the lifespan of the RBC we are taking this. So once here the lifespan is over, these are going to destroy. That time the concentration of the RBC, that means the number of RBC is going to decreases into the body. That time the kidney is activated, synthesis of the hormone erythropoietin. It synthesizes the erythropoietin. So that hormone is secreted. So this hormone erythropoietin is secreted. So because of the influence of the erythropoietin, what it is happening, sir? These are going to activate the bone marrow cells here. These are going to activate the bone marrow cells. Automatically the bone marrow cells are start over into the division of their cells and differentiating of the RBC. Their mother cells, the bone marrow mother cells are going to divide it continuously and they are going to differentiate as a RBC. So this is called as a erythropoiesis. This is the process is called as a erythropoiesis. It is occur because of erythropoietin, which is secreted by the kidney vessel. So like this, the RBC is produces in the body and they are conducting the exchanging of the gases, transport and exchanging of the gases between into the cells and the tissues. So these are the first elements. Next element we are taking that the WBC or leukocytes. The next elements, second one, so WBC here or leukocytes. So leukocytes here. Leuco means colorless here. Colorless. The name itself is explained that these are the colorless substances, colorless elements these are in the blood we are taking. So already we discussed the RBC is a major content. Then the next major ones are into the WBC. These are presented approximately 6,000 to 8,000 per 1 cubic millimeter of the number of WBC we are taking 6,000 to 8,000. So cells are presented for the 1 cubic millimeter of the blood sample we are taking. Again the WBC based on into their structure, based on the structure of WBC, they are differentiate the two types here. They are the granulocytes and agranulocytes. So granulocytes, what are the structure? What is the characteristic of the granulocytes? Means the granulocytes, these are the cells contains their cytoplasm, a granulated cytoplasm. The specific granulated cytoplasm is present in these cells. So these are called as the granulocytes. Whether the cells not having into any granules in their cytoplasm, a clear cytoplasm is present. So granules are absent. Those cells are called as the agranulocytes here. One by one we can be discussed. First one is the granulocytes. Granulocytes here. So granulocytes again differentiate based on the their structure and their staining methods. Three types here. They are the neutrophils, eosinophils, basophils. So neutrophils, eosinophils and the basophils based on the staining. So these are into the three types here. Neutrophils means the neutral dyes are staining into these cells here. So eosinophils means acidic stains are staining into these cells. Basophil means the alkali stains or the basic stains are staining into these cells. So like this we are differentiate three types of the granules. One by one we can be studied. First one is the neutrophils. So neutrophils, whether we are taking the neutrophils. So neutrophils we are taking means these are the rounded circular structures and the cytoplasm is not having into the any pigment here we are taking means, and the specific granules are presented in their cytoplasm. Specific granules are presented in their cytoplasm we are taking. And all the cell organelles are presented. Along with cell organelles we are taking the nucleus is presented, a multi-lobed nucleus is present, a specific multi-lobed nucleus is presented. So this nucleus is called as a polymorphic nucleus. Polymorphic nucleus here. So that means so many number of lobes are presented with thin connectivity. So they are forming here and the specific granules are present. So these are called as the neutrophils. Generally the neutrophils we are taking, where these are involving, the neutrophils are involving, so these are in the phagocytic cells here. These are the phagocytic in the nature. Phagocytic cell. So they are performing into the phagocytosis here. So phagocytic, and mostly these are involving into the infections of pathogen in tissues. So when the tissues are infected with the pathogen, that time their number is going to increases in the tissues we are taking. So this is a specific function of the neutrophils we are taking. So the number of granules are present in the cytoplasm and the nucleus becomes into the multi-lobed polymorphic nucleus. This is a specific character of the neutrophils we are taking this. Actually these neutrophils, we are taking the neutrophils. Generally these are total RBC number, these are the 60 to 65%. So the major component of the WBC is formed by the neutrophil, phagocytic cells. So these are first form. Second type of the cells, they are the eosinophils. Eosinophils. Actually the stain eosin, the coloring agent eosin is staining into these cells, so that only they are given the name of the eosinophils here. So the eosinophils 2 to 3%, that means total number of the WBC, 2 to 3% of the cells are eosinophils only. So these are also, whether we are taking into the structure, these are the round circular cells. The cytoplasm is having in the numerous number of the granules, minute granules are presented like that as a neutrophils, and here the nucleus mostly the bilobed nucleus is presented here. Two-lobed nucleus, 2 to 3 lobed nucleus is forming. So in the case of the eosinophils. So this is the basic structure of the eosinophils. Where these are involving the eosinophils means these are also involving in the allergic reaction. Allergic reactions and infections. So generally the number of eosinophils increases at the time of allergic reaction, that is immune reactions, we are taking, or infections. So when the pathogenic infections are causing, that time their number is going to increase. This is the second type of the cells into the granulocytes we are taking. Next one, the third type of the granulocytes we are taking means they are the basophils. Basophils here. So basophils. So basophils also a rounded circular cells and having a number of large granules are present compared to the granules of basophils. So with the other granulocytes, here the granule size becomes into the very larger and the specific here. So more larger granules are presented, and nucleus is also multi-lobed nucleus, but these lobes are not connected by the ethine tube. These are connected by the ethic connectivity is presented. So multi-lobed only, but all the lobes are connected by the ethic ones are connecting here. So this is a basophil. So total the number of basophils in the total WBC we are taking is 0.5% to 1% of the basophils are presented into the total WBC count. These are present only into the 0.5 to the 1% only. Actually, these are also involving into the infection. So these are religious into the infection, and mostly these are secreting into the heparin, serotonin and histamine. So these are the major important secretions which are releases in the infection of the person. So these are secreting by the basophils. Like this, the three types of the granulocytes are presented into the WBC. This is the first form. The second form, agranulocytes. Agranulo means so the second form is the agranulocytes. Agranulocytes means name itself is explaining that granulocytes here. That means granules are absent in the cytoplasm. So there is no specific granules in the cytoplasm. All these cells, that means agranulocytes also circular in the nature. These are into the two types here. So they are the lymphocytes and monocytes here. So lymphocytes and the monocytes. So first one, we are going to discuss the lymphocytes. Lymphocytes here. Among the WBC, these are the second largest components. Approximately, these are present the 28% of the WBC forming by the lymphocytes. Lymphocytes are secreted, are synthesized, are producing from the lymphatic tissues. So these are produced from the lymphatic tissues here. So these cells, actually the lymphocytes are based on their function. They are differentiate the two forms, B lymphocytes and T lymphocytes here. B lymphocytes are bone marrow lymphocytes here. These are developing in the bone marrow. So that we given the bone marrow lymphocytes or B lymphocytes. In the birds we are taking, birds of Africa, they are going to develop. So B lymphocytes. These are involving the immune reactions here. Immunity reactions we are taking. So what is the immunity reaction means? Whenever the antigens are enters in the body, their specific antibodies are producing from the B lymphocytes only. B lymphocytes are involving into antibody production. Specific proteins are they are secreting here. And the T lymphocytes are So thymus lymphocytes, these are T lymphocytes, means these are involving the cell mediated immunity. These are involving into the cell mediated immunity, T lymphocytes. These are helping into the B lymphocytes we are taking. So like this, the two types of the cells are presented. So in the lymphocytes we are taking. The structure of the lymphocytes we are taking means these are also circular, but the nucleus, a large nucleus is presented here. A large nucleus. The cytoplasm becomes into the very small amount of the cytoplasm only presented. In this cytoplasm also there is no granules are presented. So like this, the lymphocytes are forming. These are the 28%. Next one, the agranulocytes we are taking means they are the monocytes. Monocytes here. These are also agranulocytes. These cells are also circular and not having any granules in their cytoplasm. Clear cytoplasm is presented. The nucleus becomes into the kidney shaped, larger kidney shaped nucleus is presented in the monocyte. So these are act as a, monocytes are act as a macrophages. So monocytes are act as, so like that as a, in the tissues, mast cells. Mast cells. So the mast cell function is a phagocytic function. Phagocytic function. So like this, the mast cells, how they becomes the phagocytic. The monocytes are also phagocytic in the nature. They kills the whatever it is the pathogen or antigen that enters into the body here, and they are further converted as a, they are act as a macrophage, further converting as a macrophages here. So based on into the position, suppose into the central nervous system, microglial cells are forming from the monocytes. Suppose into the Cooper, that means suppose into the liver we are taking means they are forming as a Cooper cells here. Like this, these are going to differentiate and conducting into the function. Totally the function is phagocytic processes conducting by the, so these monocytes here. These are the two types of the agranulocytes we are doing. So finally, in the elements of the blood we are taking is that the platelets here. Platelets. Platelets. The third type of the element is the platelets, or called as a thrombocytes. So these platelets are synthesized from megakaryocytes of bone marrow. In the bone marrow mother cells, the bone marrow mother cells, the megakaryocytes are, they are going to differentiate, they are going to divide it and forming into the platelets here. So platelets, we are taking the platelet structure. These are not the specific structure. The irregular shapes they are forming. So these are look like as a broken cells, a pieces of a large broken cell here. Here the nucleus is also absent. There is no nucleus is presented into the, so these platelets here. So broken piece-like structures are forming. Other name is a thrombocyte. So what is the advantages of these platelets? Means platelets are involving in the coagulation factor synthesis. Coagulation factor. So coagulation is nothing but as a the blood clotting mechanism. The blood clot, to clot the blood. So when the injuries occurs, from that place to blood is continuously clotting, continuously bleeding. So to clot that blood at that particular injured place, they requires into the factors here. Coagulation factors are required. Totally these are present. The 12 factors are presented. So in these 12 factors are synthesizing by the platelets here. Generally these are synthesizing by the platelets. So when these are going to defected, these factors are absent, automatically it leads the So coagulation defect, blood coagulation defect, and continuous blood is going to bleeding here. So the total number of the platelets we are taking is in the person 1,50,000 to they may occurs 3,50,000 of the platelets are present. All these are the cubic millimeter of the blood sample. These are present the 1,50,000 to 3,50,000 of the cells are presented. So this is the major function of the platelets involving into the coagulation, blood coagulation, and preventing into the bleeding process here. So these are into the three types of RBC, WBC, and the platelet. Three types of the formed elements or corpuscle into the blood.